I'm recording this so people that aren't here can take advantage of this. So quickly, we should go through this fairly quickly, but there's some things I want to point out and make sure that we're all on the same page about. And I already did kind of an intro to this, right? So I kind of did this already, didn't I? But I just want to make sure that this review session is quick, and then I also want to design our final, right? I want to make sure we all know this is going to be your study guide, right? All right, so one of the things that you see here is these things. What do we call these things here? Alleles. And what are these alleles? So when we say alleles, I guess my question is, we always talk about big N and little n. What are we talking about?
Dominant recessive.
Dominant recessive. Okay, that's all. Those are all right answers. Okay, they're all correct answers. But I need you to think about what is it exactly that it is we're talking about. DNA. And it's a version of the DNA. So if I could take and look at, so this is a chromosome, right? This is a representation of a chromosome. It's a bad one, but it is a representation. There are these little sections in here that are called what? Right. They are nitrogenous spaces. That's correct. But there's these sections in here that code for a protein. So these actually do something. Not all of the DNA. So that's one fact that you need to know, right? Not all DNA codes for a protein. All right. So that's number one, right? Number two, you should know, I guess, what a protein is. And a protein, besides being a polymer of amino acids, a protein is the structural and functional unit of the cell. If you say something is done, if you say there's something doing something, that means if something's doing something, that's usually a protein. Sometimes it's RNA, but most of the time it's going to be protein. Are we clear? If it's doing something, it's a good bet that you're talking about a protein. So what makes a protein? What tells the body how a protein is made? It's this thing here, this part of the DNA that actually codes for it. That codes for it. Code means, you know what coding is, don't you? Like a program, right? If you write a program one way, you get one video game. You write a program another way, you get another video game. You know that, right? You play video games on Xbox and such. When you're playing a character in an Xbox, it has a specific set of characteristics. Those characteristics were coded by some kind of programming. This is the programming. This is the message. DNA is the message. DNA is the message that makes you. Okay? And it does that by making proteins that do things. You make one set of proteins that does one thing, you change that protein, you change what it does. So let's be clear on that. Hey, Miss Scott, come on up to the front somewhere. So, okay, so their messages, what do we call those? What do we call these areas that actually do code for protein? There are a number of them. So, go ahead, you said it. Genes. Those are genes. Genes code for proteins. So not all the areas code for proteins, but the areas that we call genes code for proteins. Do we have that so far? Yeah. I mean, we understand that, right? I mean, don't just say yeah, because we're here on Saturday. We don't want to waste our time. You want this to count, don't you? You want to just know this and move on, right? Don't you? I know I do. So you code for protein. It's called the gene. It's the part of the DNA that actually makes a protein. So we're going to talk next week about how we do that. We're going to move very fast next week. We're going to do mitosis, meiosis, and the making of a protein, both transcription and translation, as well as DNA replication. Many ideas. We've been working to understand this. It should be easy as long as you remember these basic elements. Genes code for proteins. We all got that? So what are you said, wait a minute, you guys told me these were alleles. So what are alleles?
Type.
You're right, they're genes, but what about
The type. Is that the
the type of gene? It's a message, yes, you're right, but it's a type of a gene, right? So where a gene codes for a specific trait or protein, they say eye color, let's say eye color is the gene, that's the gene. So let's say gene 1, let's say this is gene 1. So eye color, and then what?
The type of
The type you have, the type of eye color, whether you have blue, right? Let me redo that. Whether you have blue or brown, right? I'm just going to use blue or brown. What are these? The alleles. All right, these are alleles. Is everybody okay with that so far? We don't have any issues? All right, so the alleles are different kinds of genes. And so what does that mean? See, I think a lot of people, from what I hear, a lot of people understood that coming in, right? But what does that mean? That means when you have a brown gene, a brown eye color, all right, you have this. And there's many more, hundreds more.
not having this.
Right, a code, right? You have a code. The code is pretty big. I'm going to pause this while you get your donuts. So now that we all got our donuts and Gamble's going out to get his printing or whatever he's doing, you have this gene, and this part of the gene is going to be just slightly different between the two alleles. The gene starts here and ends here. I'm not going to write the whole thing because it could be hundreds of nucleotides long. I'm not going to do that, right? So I'm just going to focus on this example. Let's say brown, you get brown eye color in this dragon if you have this part of the code. Now, when you look at the dragon might have one brown eye color, it might have two versions of this, right? It could have another allele that has, because how many copies of each gene do you have? Two, right. So you have this copy of this gene, this allele is brown. So this dragon, he or she could have another brown gene, right? Another brown allele. Or it might have a blue allele, right? If it had a blue allele, it'd have the same stuff, except that maybe this happens. So maybe there's a difference in the two alleles. This is in this area, and the difference in the two alleles is that where this is a T, this is a G, and where this is a C, this is a T. So two small differences in the code means this one's blue and this one's brown. This one makes a protein. Maybe the protein doesn't work as well. Maybe the protein makes a different colored dye. Maybe that protein works better, faster, does something completely different. We don't know, right? It all depends on the protein. But in the case of eye color, we know that the brown gene overwhelms the blue gene. Okay, so we know that blue eye color is recessive to brown eye color. When we say dominant recessive, what we're saying is that if you have the brown and the blue together, your eyes are going to end up brown. And why are they going to end up brown? Because you make more brown pigment, and the blue eyes is actually making less pigment, and then the combination of the blood at the back of your retina and the light coming in, and that gives you kind of a blue color. Yes, Ramir?
So is that how some people end up with two different colored eyes?
No, that's a completely different situation we're not going to talk about. But it's a good point. That does happen. We're keeping this simple. I always hate using eye color because I'll be honest, the truth about eye colors is like something like 6 genes involved in eye colors. It's just like skin color. We're going to do a case study where we're going to look at skin color. And just like skin color, you don't have black or white, right? You have all kinds of intermediate ones. Just like you don't have blue or brown eyes. You got blue, hazel, light brown, dark brown, green, kind of iridescent. There's all kinds of different eye colors. Yeah.
So if your mom's parents have like blue eyes.
We'll talk about that in a minute. Listen, it's a good question. I'd like you to first, I need you to take this one step at a time. Too many students jump around and they want to try to understand everything at once. It doesn't work that way. So first understand that here's the question, do you understand that you have a gene, that gene is located on a chromosome, that you have 2 copies of that gene, that every version of the gene is an allele, then an allele can be dominant, one allele can be, doesn't have to be, but can be dominant over another one.
Wait, so can you have 2 dominant alleles?
Yeah, you can have 2. So if you have 2 brown, so where are you getting these 2 alleles? So these are 2 chromosomes, 2 copies of the same gene.
The X or Y?
One came from mom and the other came from dad. So can you have 2 brown ones? Sure. If your dad gives you a brown gene, your mom gives you a brown gene, you got brown. You only have brown. They only have the brown allele, but you only have what you get. Does that make sense? That's going to be very important moving forward. You only have what you get. You don't have anything else. Yeah.
So you're not people who like get their eye color changed? Yeah.
Okay. Can we not see? I love you, but that's just we're going off in a tangent, and I'd like to stick to where we're at. So. These genes, these alleles came into the cell. Does that make sense? But from mom, one from dad. They could be blue, they could be brown. And as I said, there's actually 6 or 7 of these, so maybe it's more than one gene, see? The thing is that at this gene, there's only brown or blue, right? But there's 6 other genes that control this. So what you got to understand, and it's difficult to understand it at this point, but you'll see, I hope, before the end, is you got this machine, right? And this machine has parts. And there's all kinds of funky shapes and parts to it. And how they all interact with one another. They all interact with one another. This part influences this part, that influences that part, that gets influenced by this part, that influences that part, that also influences this part. And you see they all it's a complicated machine. And we're talking about the gene, this part right here for this gene for eye color. But you know what? This gene also has something to do with it, right? Right? Because it's part of this machinery. And so does this gene, and so does this gene, and so does this gene, and so does that gene. So things get complicated in reality, and hopefully you'll understand a little more as we go along. But as we make these proteins, each protein then does something that influences something else. It's like you have this complicated machine, right, and you take one piece out. What happens in the machine?
It won't work.
It won't work. But if you change one piece of the machine, who knows? It might work better, might not work at all, right? So the machine is complicated, but to keep it simple, trying to keep it simple, what we can do is just say, okay, look, we got dominant recessive. Let's just think of it that way. When you have this version here of this gene, you get brown eye color. When you get this version of this gene, you got blue eye color. For right now, that's what we're going to say. And because every time you have this version, you get brown, even if you have a blue, right? You have the blue. This person has blue, right? Has a gene for blue. Which one do you see? The brown. Why do you see the brown? Why is the phenotype brown? Because it's dominant. Because whenever you see, when you have this gene, this gene makes so much brown pigment that it doesn't matter that there's a little blue in it. All right, this is not how it works, but let me go ahead and give you an example. If you have a bunch of brown paint and you have a little bit of light blue, you only make a little light blue and you put a lot of brown in there. what color are you going to get? All right. So in this, when we're talking about dominant recessive, we're talking about, does it make a lot? Does it make a little? Is the stuff that they make, is it better? Right? So that's what we're talking about. We're talking about genes and alleles. Does everybody get that? Because when you look at this, and you did this in middle school, some of you, and you look at this, it's very simplistic, isn't it? But what you got to understand is every time you see a big N, you got to think this, some kind of genetic code. And you see little n is a different genetic code. And big N in dominant recessive, this is just one type of inheritance. You'll see over Christmas, over winter break, you'll see that there's a bunch of different kinds of inheritance patterns. Yeah, you have a winter break packet. So you have a capital N low. So dominant recessive just means that you have a dominant form and a recessive form of the gene. So if you get the dominant, you're going to get what? A long neck. If you have a recessive form, what do you get? If you have a dominant and a recessive together, what do you get?
Dominant.
Long neck, the dominant. If you get two big N's, what do you get?
Long neck.
And if you get little n,
Short N.
and that's it. All right, but you have to always remember that these symbols mean all this. Is that clear? All right, so let's go ahead and answer these. I think you got this. I don't think I need to write. Do I need to go through this? No. I think obvious and pretty straightforward. This is a piece that people got confused about. They said, what do I do about the genetic code? Look, you're supposed to come up with your own, right? Randomly by flipping a coin, right? You could, if you have yours, could you somebody tell me the genetic code that they chose with flipping the coin?
Here.
Oh, you got it here? Thank you. So this person got this code. Capital N, little n, big H, little g, big G. You see, I can grade these easily because everybody's code is going to be different, right? If I see two people with the same code, what am I thinking? Yeah, what are the odds that somebody flipped the coin just at the same way? It's highly unlikely. If they did this, they wasted their luck because they should have won the lottery, right? Because this is about the same odds as a lottery, more or less. All right. So, anyways, this code represents a living creature, doesn't it? This code actually could describe a creature, right? I have the list here. You guys can reference back, and this is beautifully done, by the way. Who gave me this? Yeah, very nice. All right, so we got long neck or short neck? What is it? Long neck and horns, right? And we got, is it green or gray? Green, because you have one capital. As soon as you see a capital, as soon as you see a dominant, that's what you see. The little ones, what do we get?
No spikes.
No spikes, and we have one big T, so what do we get? Three toes, and one big B. Black tail spikes, and little f. Not fire breathing. Little I, big I.
Long tail.
Is it long tail?
No, that's L.
Oh, that might have been L. I thought it was an I, but it could have been L. Is that an L? Okay.
So what's with the big?
So that's little L, and I can't erase, so big L, little L. So what do I got? Long tail, okay.
You got red wings.
And you got red, we have, big R is red wings, and little W is white belly, and big K's are freckles, and big E is red eye. So this actually describes a dragon, and she was able to make it, and let me see if she did a beautiful job. And she did do a beautiful job, and here it is. You can see it. I can't show it. I'm not going to show it to you on the screen, but there it is, beautifully. She cut out her dragon. I like it. She's a pretty dragon, or he. Is it he or she? You didn't do the X or Y chromosome though. Did you get an X or Y? You made it a he. So what did he get?
He got free.
So he had an X.
He got the ear.
He got X from mom, and Y from dad, and that means he had ear frills, and that's why he has ear frills, and so there it is. So this is the guy, and okay, so now we look at the genotype for each of these, and you fill them in based on your code. Now this is chromosome what? What does it say? What does it say? Chromosome what? Pair, right? Chromosome pair, because every chromosome has how many? 2. So we got two times this chromosome number. We got these different traits. Each trait here. So I'm not going to write all of the different genotypes and homozygous, heterozygous, or phenotypes, but I'm going to write a 1 or 2. And based on what is written up at the top, all right, so with neck length, we have big N, little n. That means, what can this father give his children? No, this father can give his children either a big N or a little n. So you got to understand that, okay? And then, is this homozygous or heterozygous? It's hetero. Hetero means different. And what's the phenotype? Now we want to say long neck. This is what this parent has. All right? This parent can give either one of those genes. Is that clear? Now, eye color. What are we dealing with eye color? What eye color do we have here? We got big E or little e. So this pair can give either a big E or a little e. And that is this homozygous or hetero?
Hetero.
And that's what?
It's red.
Red. So I'm not going to do the rest of these, but that's what you would do, right? And where are you getting that information? This is feeding all the chromosomes. Now please notice that where is the gene for eye color? Where is the gene for eye color? On chromosome 1. It's always on chromosome 1. It doesn't jump around. There are genes that jump around in corn, for instance, but not in humans, not in dragons, right? Huh? In corn. In corn. It's a whole different set of genetics. There's advantages and disadvantages. We're not going to talk about it. But it just let you know that it's not always the same way. Life does some things different. Anyways, in the genes that this creature has feed into what it becomes, but these genes are on specific chromosomes. On chromosome 2, you have what genes?
Tail length, body.
Tail length, body color, color, wings, number of toes. You always find these genes on chromosome number 2. Always have these genes on chromosome number 2. All right. And these 3 genes are on chromosome 3. These 2 are on chromosome 4, which chromosome 4 is, looks like chromosome 4 is the sex chromosome, right? X or Y.
Can I
All right. Huh?
Can I ask you a question?
Yeah, go ahead.
So, my mom's parents, right? They have blue eyes, right?
We don't want to talk about that right now, because we have...
Because I'm thinking about it too much now.
Okay, go ahead.
And then my mom has brown eyes and my dad has brown eyes, but then they made a child and he has blue eyes. Can they do that?
That's the definition of recessive, right? That's the definition of recessive. We'll talk about that in just a second. All right, so to answer that question, I really would rather wait till we get to the Punnett squares, if you don't mind. All right, so this is a male, right? So this male is going to make some sperm, and it's going to make a sperm is going to look something like that, and it's going to make a lot of them. And half of his DNA is going to go into this sperm. Does that make sense? Yeah. So if you're looking at neck length, let's say, how many different alleles does he have for neck length? 2. He has 2. Do you see that? He has 2. So he's going to put one of his ends for neck length is going to go here, and the other one here. Now pay attention, because this goes to talk about your blue eyes. The female of this dragon's mate, is going to make 2 eggs. If that female is also heterozygous, so that female is big N, little n, right? Do you see that? And these two are going to come together. Which combination are you going to end up with? It depends. It all depends. Which sperm hit which egg? Right? If big N here goes with big N there, you get the dominant trait, right? If the big N here goes with the little n there, it goes dominant. And if it goes big N, little n, it's dominant. And if it ends up little n, you have the recessive.
So it can only hit like one gene, only with one gene.
One sperm is going to meet up with one egg.
They can't meet up with like both.
One sperm meets up with one egg, and that's it. If you get 2 sperm coming to one egg, you got a problem. Cache, you cannot sit there. So, one sperm meets up with one egg. That's it. Cache. Over here. All right, so let's see if we can make better heads or tails of this. So this was what you were supposed to cut up in order to make your dragon. It was done well. Then you're supposed to paste it here, right? And then here are your chromosomes. You're making a chromosome. You're only giving one of each of those chromosomes to your child. So the male, you were asked to put one, right? It says you flip a coin and determine if you get one, which allele you get at each of the genes. You'll notice that these are the genes on that chromosome. The sex gene is found on the 4th chromosome. So each of these genes, you have a choice. You're either giving the big N or the little n, the big A or the little a, the big T or the little t. That's what you're doing. Is that clear? Are we good?
Yes, sir.
Go ahead and get water. You got real lucky, Miss Smith, that it wasn't recording while you were talking. Hopefully your mom watches this and realizes that you do talk when you're supposed to be listening. And then let's say this is an X. Now this is the chromosome, this is one copy of each of these chromosomes, right? That father can give. That's what he's going to put in each of the sperm. So if he's making this sperm, one of each of these is going to go into that sperm. 4 chromosomes, one copy of each. Does that make sense?
Yeah.
But remember, he has 2 chromosomes, so I'm going to make another set here, which is not part of this activity, but you should understand that he also has another set of chromosomes, right? And that other set of chromosomes, in that he'd have a little n, a little e, a little h, a little s, a big L, a big G, a big R, and a big T, and a big W, and big B, and big K, and you have big F, and a Y. You see?
There we go.
So this, he could give either one of these. Which one goes in there? Which one goes into the sperm? It's random. So which of these chromosomes go into that sperm is a random event. So you have 1 million sperm, and you can have any combination of these genes, of these chromosomes. Is that clear?
Yep.
I mean, am I making sense? Okay, so when we're making the baby, which you had to flip a coin. You weren't supposed to put both copies into one chromosome, because each chromosome only has one copy of each gene. You see, if you made that mistake, you didn't understand what we just went through. And if you still don't understand what you went through, it's because you weren't listening, right? So I want to say this again. On each chromosome, you have a specific gene that you always find on that chromosome. If you have a different version of that gene, it's found on on the second copy. You have 2 copies of each chromosome. Is that clear? People are nodding, but I suspect that there's a lot of confusion still. So then you get together with your partner, and this is your partner's data. So in this particular section of the Dragon Genetics Worksheet 5, this is where each of your partner and you combine your chromosomes. So your chromosome, you might give a capital N, but your partner would give a lowercase n. You might give a little e, but your partner gives a little e. So each of these alleles are, one half is coming from the mother and one half is coming from the father. One chromosome comes from the mother, one chromosome comes from the father. And so again, here you would, after you come up with the genetic combination, you'd find out the phenotypes. Then you paste, and that's it. That's your baby dragon. If you understood what was going on, you're good. Now I'm gonna say it again. And then I'm gonna answer your question.
Okay.
All right. Father is gonna give is two possible sperms Dad could give. And that could be, that father could have a big H, depending on what the father has. Okay, what did I say? Wow. So the father can produce two types of sperm, depending on which one of the 4 chromosomes he has in the DNA of the sperm. So if Dad, and this sperm might have a little h, this sperm might have what? The big H. So when we translate it to a Punnett square, we say big H, little h. Do you see that? But it's all about, what is it about? Which gene, which sperm actually came into contact with that egg? Right? Was it this sperm that had the big H? Or was it this sperm that had the little h? Who knows? That's why we do probabilities. That's why we're doing, what's the probability of getting, blue-eyed or long neck or short neck or wings or frills or what have you, right? Because we're not sure. We can't tell which sperm is going to match which egg. And so the same thing goes with Mom. Mom has eggs, though. She doesn't have sperm. So she has eggs, and maybe I should do a pink or something. I don't know. So Mom has these eggs, all right? But she also has these 4 chromosomes, doesn't she?
Yeah.
She has these 4 chromosomes. She has an XX, so both of these are X, right? She has an X here and an X here. Dad has an X on one and a Y on the other. Remember that. But this is called a monohybrid cross. So here we're only dealing with Punnett square that's only dealing with one gene at a time. Over winter break, you're going to be dealing with a Punnett square that deals with 2 genes at a time. All right. So this is HH. Mom could give, maybe Mom is, what is H again? Horn, right? And big H is horn, or is little h horn?
Big H horn, little H horn.
All right, so little h, no. So Mom doesn't have any horns, let's say, in this example. Dad does have horns, okay? Well, what's the probability that the children are going to have horns?
25.
50%.
Well, let's see. Big H, little h, little h, little h, big H, little h, little h, little h. So now we say it's 50% chance that you're going to have no horns and 50% chance that you're going to have horns, and that's it. But why are we talking about chances? Because we don't know, we have no idea which of these 2 eggs are going to match which of those 2 sperm. It's all random. But only one egg goes with one sperm. Now let's talk about Lean's problem. Lean has a problem that she wants us to try to figure out to help explain to her. Her grandparents had blue eyes. One of her grandparents had blue eyes, yes?
Yeah.
All right, so we said blue eyes is recessive. Now pay attention. So her mom, who was a grandparent, mother, or grandmother, or grandfather?
Mother.
All right, so grandma, the female, had 2 eggs, right? Remember, dominant is brown, right? And recessive is blue.
But she had 2 little B's. 2 little B's.
That's right. Grandma had 2 little B's to give. Okay? Now, what was granddad? We don't know. But we do know he had big B. Did all his children have big B, brown eyes?
No.
One of his children had blue eyes?
Wait, what are you talking?
Your grandmother had blue eyes.
Yeah.
Your grandfather, did he have blue eyes? Any of his children have blue eyes?
No.
They all had brown eyes.
Yeah.
All right, so how many kids did he have?
You don't want to know.
A lot. And they all had brown eyes. So it's probably true that he had big B, big B. Let's go ahead and go with that. Are you paying attention?
Yeah.
Okay, so he had big B, big B. So what are all his children going to be, Lean? They're going to be brown, but guess what? They're also all going to be what? They're also all going to carry what?
Recessive.
They're all going to carry the recessive. They all have the gene for blue. All his children have the gene for blue, even though they all have brown eyes. We call this being a carrier, right? We call this being a carrier. They have the recessive gene. That's what recessive means. You got it. You got the gene for blue eyes. But you got the gene for blue eyes, but you don't see it because you have brown eyes taking over. Do we understand that so far?
Yeah.
And now you're telling me that one of these kids, it's your mom, or is it your mom or dad?
Mom.
So your mom married your dad. You said your dad had a kid with blue eyes?
So like, no,
Did-
that wasn't my question. Like,
Huh?
so if my parents both had brown eyes, how does the kid come out with blue eyes?
Okay, listen and watch. I'm asking you a question. Did your mother and father have blue eyes?
No.
Did your mother and father have a child with blue eyes?
No.
Then why are you asking this question?
Because I want to know
You want to know what?
how they come out with blue eyes when their parents have brown eyes.
Just some random person? How did they?
Yeah.
Okay. I thought you had a brother with blue eyes. That's what it sounded like.
No.
Well, let's say, what color do both of these parents have, Lean?
Brown.
Right? Lean, what are the chances that these parents are going to have a child with blue eyes?
25%.
25%. So why do you ask me that question? Don't you see that you have the opportunity that 25% or one-fourth of their kids are going to have blue eyes? Huh? I can't hear you.
I said never mind.
Okay, so it makes sense to you now? Okay. Yeah.
Since you got a big B and a little b, don't they consider that being a hybrid?
Well, no, not hybrid. We call these heterozygous.
So what do they consider a hybrid? The last year only did they had a hybrid.
Can we talk about that next week? You guys are jumping around. This is the problem with a little bit of knowledge. People with a little bit of knowledge end up confusing themselves all the time. All right? You can't assume that you understand the situation. Does that make sense? You're asking what's a hybrid. We'll figure that out. What did I say, Raven, Ramir? What did I say? You don't recognize you were just having a conversation with Raven? Did you recognize that you were just having a conversation, Raven, Ramir? You did not realize you were having a conversation. Okay. All right. So everybody understand that?
Yes.
All right. So now we're ready to make the test. You want to start with making the test? All right. So we're going to design the test together. And we're talking about your final. And as we're designing it, we're going to add what are the quizzes going to be. Because have you read your Jupiter Grades messages? Who has not read the Jupiter Grade message? I'm going to pause this. You're going to get online and read your Jupiter Grade message. So now that we cleaned up that mess, we spent a good, half an hour cleaning up that mess. Now, let's go down and focus on what we're here to actually do. Is there anything that confused you before we go on with the two experiments? And if I don't think I answered it on the video, I want to make sure I cover it now. What were the two experiments? Who knows what they were?
The argument in
That's right. The Griffith experiment, which dealt with what was the hereditary molecule, right?
Yeah.
And they had three choices. It was either protein,
DNA.
RNA, or DNA. And you can do the reading, and you can do the
What changed? What was the transform?
right. So what was the hereditary molecule? Another way of saying that is what was the transformational molecule, right? What was the thing that changed the bacteria? And that led to understanding what was a hereditary molecule. That's it. So that, and there was all kinds of steps to that. Everybody understand that so far?
Yeah.
We all understand what all the steps are. So what do you need to know? I already said this in the previous video, but I'm going to double check you get this. You have to know the steps. You have to know what are the control groups. Right? And you have to know what did the experiments show? With a focus on what? With a focus on the logic and techniques used. Okay? With a focus on the logic and techniques used. Are we clear?
Yeah.
That's what you need to know from the Griffith experiments. The video goes into detail. The first video that was through all the answers, all the steps, I walked you through it. Then the second video that I sent you last night summarized it, and then we went on with the second experiment, which was what? Meselson install. Right? And the Meselson install experiment did what? How is DNA replicated? That's right. The experiment, they used nitrogen 15 and 14. They used a density column that grew bacteria on two different plates. The video was extensive. I added the little extra videos from YouTube. I downloaded them and put them into the video myself last night, and then uploaded it. And you had kind of three choices here, right? Just like you had three choices up here, you had three choices in this experiment as well. And the first was semi-conservative.
And demo.
That's right. And conservative. And the third one was dispersive. All right, so we had those three choices. And again, I went through all the steps, all the background knowledge. You have to know all that. You have to know all the steps, all the control groups, all the different, what did the experiment show? And then the logic of each step, and the technology or the technical aspects, right? The density column, the petri dish, growing the bacteria on nitrogen 15, then growing it and transferring it over. Why did they do that? How did that make sense? Why, one thing some people were confused about, and I'm going to go ahead and talk about it here if you don't mind. One thing people confused about was the difference between the three models, model 1, 2, and 3, right? Which was, again, semi-conservative, conservative, and dispersive, right? Those three models and the density column. They got confused. They got confused about what was expected, right? And why was that the way it was supposed to be expected. I don't want to go through that whole experience again. It's all on videotape. I'm going to tell you, go ahead and watch the video. I think it went through step by step. I don't know what else to do. You have to kind of watch it, break it down. But I'll say this much. the semi-conservative model predicted something. If you grew, that's if you grew bacteria on a plate, and I mean petri dish, right?
Yeah.
With nitrogen 15 and then transferred to nitrogen 14. If you did that, the semi-conservative model predicted something. The conservative model predicted something. And the dispersive model predicted something. What did they predict? Go back and look at the video and ask yourself, what did they predict and why did they predict it? I explained it. Maybe you kind of zoned out in class, or maybe you kind of missed it, but you have an hour and 15 minutes of video. The YouTube is waiting in your Jupiter grades. You don't have to watch the whole thing. Go to the part where you're confused and really listen, because I'll walk you through it. All right? So there's these density things, and if you took that DNA, if you waited after one round of replication, and this is something that some people get confused about, and then round 2 of replication. So every time these bacteria, they start off here with a certain DNA type, right, with a certain chromosome that has a certain density, and after you transfer it to the nitrogen 14 plate, it goes through one round of replication. What does each of these models predict? And then it goes through another round of replication. What does each of these models predict? I answer these questions in the video step by step. You have to know that. That's definitely going to be on the test. Okay?
Yeah.
All right. So that being said, we know. Let's go ahead and design this exam, this final exam. And then let's talk about what the quiz questions are going to be like on Tuesday. They won't be on Monday, they'll be on Tuesday. So the first things, of course, are going to be the Griffith experiment. How many questions do you think are going to be on the test by the Griffith experiment?
6.
3.
I'll take the average, I'll say 3 questions, all right? 3 questions. All right, then the second experiment, and they're going to be good questions, they're going to be in depth, they're probably going to have the whole experiment, and you're going to have to, and there'll be pieces and parts missing, and then you're going to have to answer some questions. Or maybe it'll be a different experiment that's like the Griffith experiment, but it uses the same content, the same ideas, and you'll have to figure out what the answer is. Then you got Meselson and Stahl. I'm going to put M and S. If that's okay, I'll put M and Stahl. How many questions do you think I'm going to ask from Meselson and Stahl?
4.
I'm actually going to ask 4 questions from Meselson and Stahl. Now I can put up to 100 questions on this final. Up to 100 questions on this final. It's going to take you 2 hours, and that's what your final is, 2 hours long. So don't expect. I've never made a test with 100 questions on it, but I could. So that's what we have to work with. Either you're going to complain or you're going to help me work. You're going to help me design the final. Which do we want to do? We could go outside, break some glass, and walk around and talk and complain, or we can design this final. All right, so 4 questions. So your first quiz, what do you think your first quiz, quiz 1 on Tuesday, covering these 2, how many questions do you think I'm going to have on that quiz? Let's do 6.
I would say 5. Yeah, 6.
You say 5?
Yeah. I could do 6.
5 questions sounds good to me. 5 question quiz on Tuesday. And what Tuesday am I talking about? I'm talking about Tuesday, December. 7. Is it 6 or 7? I think it's 4, 5. Yeah, 6. Right, so December 6th. We're good with that then?
Yeah.
So what else am I I'm gonna look at DNA structure, which you should already know. And I can promise you that part of your problem right now in understanding the Demesel-Stall experiment, part of the complete confusion some people have is they don't know their DNA structure. All right, so DNA structure. And what am I talking about? Look at the videos I gave you. We covered it in class. Look at your notes. But you need to know your DNA structure. And of course, there you're talking about double helix. What does a nucleotide look like? The histones. What is it? Chromosome, chromatid, sister chromatid, chromosome, chromatin. Where is the DNA found? Right? All that's going to be on there. And I would say there's going to be about 6 questions on DNA structure on this exam, on this final. The next quiz, I don't know. I think considering that you have only 2 weeks before the final, I think a quiz every other day is a good idea. So we'll have a DNA structure quiz on Thursday, and that'll be quiz 2.
Thursday.
And that'll be December 8th. And that'll be 3 questions.
December 8th.
Yeah, it's on the board. So So DNA structure is covered there. What else did we cover in class that we need to have on this test, on this final? Early Earth. That's a lot of information, early Earth. It's a few days. We know you need to know what? You need to know the atmosphere.
How many questions?
You need to know the atmosphere presently. You have to know what changed it. You have to know what was the first life like? You have to know endosymbiosis. You have to know what is photosynthesis. What is cyanobacteria? All right. What are microfossils? It's a lot. I would say about 20 questions on the test are going to be from all this. I think that's fair. Spent a lot of time on this. All right. What is that? What idea? Can life change the earth? What's interesting is that not just life, but the smallest of life, the most insignificant supposedly of life, can change the earth. Photosynthesis, yeah. What does photosynthesis have to do with it? Is that better? All right, so 20 questions here. So I think it's fair, since we've already covered it, we went over it so much, that we go ahead and do a quiz on Friday on this. No, we might have Friday meeting. I'm not sure what's going to happen on Friday. So what might we do here? Then the following Monday. The following Monday, which will be the 12th. That'll be review week, right? The 12th. So Friday, or Monday, December 12th. These are quizzes. These are going to count for points. And how long are we going to make this quiz? It's a Monday. There's 20 questions for on the test. So I think 10 is reasonable, but I'm going to go ahead and say 7. You know what? I'm going to do 5 because I don't want to waste my questions. There'll be 1. Now, that takes us all the way up to where we are now. Oh, wait, no, it doesn't. We still have a couple more things. We have the dragon genetics, right?
Yeah.
Can I go on?
Yeah.
How many total questions do we have for the test so far? We have 3 for Griffith, 4 for Stahl, 6 for DNA, and 20 for Earth.
33.
So 33 questions. So we have 33 questions. We can go up to 100. So part 5 of this final then has to be the dragon genetics. Right, dragon genetics packet. Now this dragon genetics packet, what did it cover? It covered chromosomes, genes, phenotype, genotype, dominant, recessive. What else? You girls know you're having conversations, right? I just want to check in on you. I'm not sure if you knew that. Chromosomes, yep, DNA / alleles, right? The difference between not in dragon genetics, but that's interesting. We'll talk about it next week. So, huh? Yeah, there's a lot of stuff you're gonna have to review, huh? A lot of stuff. That's why we are making the study guide right now. You're making the test. Everything you say will be held against you on the final. Homozygous and heterozygous. All right. So that's how many questions on this part, from this part of the test?
At least 6.
I would say 10. 10 questions on this, 10 questions. I think you guys are solid on this. I really would not want to waste quiz time on it, but if you need some practice problems, there will be some practice problems for you on Monday. They'll be waiting for you on Monday for a lot of this. And it'll be kind of a review sheet. Number 6, let's not forget membranes. Yeah, this is the one thing you were hoping I'd forget. And I'm going to include in that cell structures. So you better know your cell structures. Yeah, the nucleus, the endoplasmic reticulum, the structure, the function. You better know it. And you better know osmosis. You learned it in my class already. Diffusion osmosis. You better be able to tell me where the water is moving, if it moves, where the water moves. If it moves. Because when I tested the membrane after school with some kids, they were confused on whether water moved or not. So you better be careful. The different kinds of proteins, membrane proteins. Right? membrane proteins, that means the channel proteins. Active versus passive transport. Within passive transport, you have diffusion, and you have facilitated diffusion. If you've forgotten any of this, it should be in your notes. If you don't have it in your notes, you have the videos still waiting on my YouTube channel. What was that? Oh, by the way, DNA structure. It's just an aside. Let me put it in red. Remember, P.S. Remember, with DNA structure, don't forget Chargaff's rules. The percent A equals percent T, percent G equals percent C. And so if I give you one, you should be able to calculate the other, all the others, right? So don't forget that. And that, again, in the video that I posted, it goes through an example. And that takes us to membranes, it takes us to organelles, it takes us to dragon genetics, it takes us to, which is simple Mendelian genetics, but we're still going to go over some more of that and go into some more complicated stuff. Oh yeah, good question. What about the membrane? I think 15 questions on the membrane. There's a lot here. There's a lot of ideas here that need to be tested. So 15 questions. So right now we're up to how many now? 45, 55, 56, 57, 58 questions so far. 33 from the previous section. So 45, 65, 67, 68. 69, am I counting wrong? 30, 40, 55, 56, 57, 58. 58 questions. 58 questions so far. So we have 58 questions. Again, we can have a maximum of 100. So you can expect to see, you can see 100 questions on a 2-hour exam. All right, so we still have to cover some stuff next week. So starting Tuesday again is what date? December what? 4, 5, 6, that's right, 6, thank you. On Tuesday, December 6th, we're going to cover DNA replication.
After the quiz?
That's right, after the quiz. The quiz is only going to be a 5-question quiz. On all the quizzes, it's going to be a quick 5-question multiple choice quiz. The quiz?
Remember you said it was going to be 40 questions.
No, did you read your Jupiter Great message that I paused the video, the recording for and asked you to read it? No, you did not. I said it.
She didn't.
Oh my God.
I asked her, that's what we were talking about.
Can we go over it? But she didn't understand it.
I'm trying to pass the quiz.
Did you read your Jupiter Great message?
She had her and somebody had this. That's what I'm here for. So
Okay, so DNA replication, boys and girls, on December 6th, on Tuesday, after the 5-question quiz. For those of you that were in 6th period, especially those of you that were absolutely guilty of talking and causing the mess to begin with, I don't remember which one of you was, the two look alike to me, so I can't tell. Those of you that are guilty of causing the problem in 6th block, one of the main conspirators of the talking ring. Treason is what I would call it as well. You guys, at the end of that class, I told you you'd have a test on Tuesday, right? Yeah. Did you read your Jupiter grades? So do you have a test on Tuesday? No, you don't. It'll be a quiz with 5 questions. because I decided not to let the few people mess it up for everybody. That would be you, Miss Smith.
You asking questions on the teaching. You keep on saying I was talking.
Maybe, here's what I don't get, guys. Here's what you don't get. You guys have to understand this, and for some reason you're not getting it. I understand that you're not talking about nonsense, although sometimes you are. What you don't understand, children, is that I'm trying to explain something that's very complicated. That any little piece that you don't understand is going to get you a wrong answer. And I want you to get the right answer. So I'm trying to get you to understand each and every little piece apart, which sometimes can lead to a long chain of explanations. And that means, like any good chain, every link counts, right? Every piece counts. And if you're busy talking about what happened yesterday in science class, if you're busy talking about, I don't care what good thing, what reasonably important thing you're talking about, you missed this link, you messed up this entire section on your test. That's what I'm trying to get through in your head. You got to pay attention. The excuses, there's all kinds of excuses. All right, DNA replication is going to be we're going to cover it on Tuesday. That is a lot of detail about, and here you're going to have to know the enzymes, the important enzymes, where it happens, and there's only 2 enzymes. There's going to be 2 of them that are going to be really important, but you're going to have to understand how the process happens. Watch for the video, dear. It's going to cover the whole review. All right. If you have any input, you really should give some input now on how many questions or what content that you know that you want in on the test. Right. We're out of the stuff that we already covered. We're talking about the stuff we're going to cover next week. Okay. How it happens? So where it happens, how it happens, and of course, when it happens. In DNA replication, you can't talk about DNA replication unless you talk about the cell cycle. You can't talk about, and that there's several parts to it. The parts of the cell cycle are going to be important, and how much time, how long it takes to get through the cell cycle. And then, of course, the last thing you're going to be able to talk about here with cell replication is mitosis and meiosis. My hope was that we would be able to do protein transcription translation. We will do transcription translation, but I think that's going to have to, that might have to wait for as a winter package section, and then for us to go through it when we get back from winter break, because I think this is a lot. I'm being overly optimistic if I think I can get much more out of you than this. All right, so mitosis and meiosis is the steps. We're going to have a lab. You're actually going to pull out the microscope, so you're going to look at mitosis and meiosis under the microscope. So there's going to be some work to do this week. Remember, this coming week is not the week before finals. We still have another week of review after this week coming up. So the steps, what does it look like?
Can you put this video on YouTube?
I am, dear. That's why I'm recording it. What does it look like, right? What does mitosis and meiosis look like? What is happening? So you're going to have to be able to identify the steps of mitosis. You'll find this actually kind of fun, and it's actually easy. What is happening at each step? The purpose of each. What's the purpose of mitosis? What's the purpose of meiosis? And for meiosis, and this is kind of an aside, it's a PS, right? PS for meiosis you need to understand why is it important. And what is crossing over. What is crossing over? What's happening? Why is it happening? All that. Cell cycle, DNA replication. You have to, of course, you'll know, we'll cover mutations. Right? Why are these important? Why are mutations? Why are they important? How are they important? What are they, rather? The different types of mutations. So that's a lot next week, right? We spent a lot of time this week talking about DNA structure. We spent a lot of time this week talking about the different models of DNA replication. Do you know why we spent all that time doing that? So you can understand this. All right, so we're going to go through a lot. We're going to go through a lot this week. And you're going to have, let's be clear on this. I want to be clear. You're going to, there's some of the, let me go ahead and reduce this a little bit. There's some to-dos today, this week. This week, the week of December 12th, you're going to be given review questions for practice, right? That's your quizzes and some homework if you want. The homework is optional if you want to do it. I would suggest you do it. So December 12th, you're going to get that. You're going to have a lab, mitosis lab. And you're going to have an activity. Mitosis lab and activity. And you're going to have a virtual lab. You're going to be very busy this week, aren't you?
Yes.
So it's going to be a lot of work. You have to do this and review. Don't wait till the following week for review week to start going back over your notes, right? Or to come after school, because remember, on Wednesday and Friday, I'm going to be absent on Monday. I have a doctor's appointment. But on Wednesday and Friday, you can come after school and get some tutoring on this stuff, okay? How many questions do you think I'm going to put on this test concerning all this? 25 questions. 25 questions. So someone add 25 and 58? 83? Are you sure? I'm going to put it down.
Hold on, wait, what?
25 plus 58.
83.
83. I hope you're happy, because what we're saying here is you're going to have 83 questions on your final. 83 questions on your final. You just went through how many you're getting in each of the topics. You know what's coming on the final. You should start reviewing now. If you don't know your organelles, you better go memorize them. What are the steps that you need to do? Let's be clear. You need to be revising your notes. Remember, your notebook is being collected on that on that Tuesday of finals week. Number 2, study groups. Number 3, tutoring. 4, videos. 5, practice problems. And 6, Quizlet cards. Or index cards, any way you can to recite this information. Memorize what you need to memorize, but understand the logic, so when you see a question that's not exactly like the one you were asked before, which will probably not be the case, then make sure that you are ready to be able to adapt and apply your knowledge. All right, that's it for the review today. I need to print out some review questions for you in your review packet. So I'll be doing that until 12 o'clock. If you have any specific questions, you can stay. Make sure you sign out. I'm going to take a picture of, thank you. I'm going to take a picture of and send a copy of the sign in, sign out to your parents. All right, have a good day.